Thank you. Yeah, so my name is Dr. Stephanie Swift, and as you just said, I'm a scientist in the lab of Professor David Stojdl at the Children's Hospital of Eastern Ontario Research Institute right here in Ottawa. And I'll be talking to you today about some of the basic biology behind brain cancer and how that feeds into current and innovative new treatments that are being developed. And I'll talk a little bit about one of those treatments that we're developing in our lab, at the Stojdl Lab, which is using viruses to treat and kill brain cancer. So there are two types of brain cancers. They segregate into primary brain cancers, which arise directly in the brain itself, and secondary brain cancers, which arise outside the brain, elsewhere in the body, so for example in the liver, or the breast, or the colon, and actually spread into the brain, and that's called a metastatic disease. It spreads from the outside of the brain in. And in terms of primary brain cancers, there are about 2,500 new patients diagnosed in Canada each year, and around half of these are of the subtype gliomas. And about 50% of gliomas are of the particularly aggressive type, glioblastoma multiforme, or as Denise said, multiforme. The median survival for glioblastoma multiforme is about 14 months, and despite a lot of intensive research over the last 30 years into brain cancer research, we haven't really been able to do much to change this median survival time and improve it too much. One of the best innovations of the last 30 years is the specialty chemotherapeutic temozolomide, which is helping people to live longer and improving their lives. So that's a good thing, but there's definitely a tremendous opportunity for new therapies that can improve these kinds of things that we use for existing treatments even further. In terms of secondary brain cancer, where it spreads from the outside of the brain in, there are about 7,500 new patients diagnosed per year in Canada alone. So slightly more than the primary brain cancers. It's definitely a greater challenge for the Canadian healthcare system. And in fact, brain cancers themselves are a leading cause of solid tumour death in children, so they're a particular challenge for paediatric oncology. So what makes brain cancers so difficult to treat? And the answer to that is based on a huge number of factors that are really inherent to the brain as a specialised organ in the body, that you don't find these factors in other organs outside of the brain. And so I'll just take you through a few of those reasons now. One of the first difficulties is the fact that brain cancer is an infiltrating disease. So despite the fact that the main tumour mass might be a nice sort of tight circle here or here, we actually find that the brain cancer cells themselves are incredibly invasive, and they like to spread and go to new areas of the brain. So you can see that the brain cancer cells spread in these sort of tendrils that extend out from the main body of the tumour or extend into surrounding tissue as well. So that makes surgical resection very difficult, because when a surgeon goes in and tries to take out the primary brain tumour, that's fine, it's in a nice little mass and they can access it very easily. But what they can't do is access these sort of patches of disease that spring outside the brain, the main tumour itself. And that's because the brain itself is an incredibly complex structure. You have millions of neurons all feeding different areas of the brain and involved in things like cognition, learning, language. And you can't do too much damage to these areas, because otherwise that would be very detrimental to the patient. So a surgeon is very restricted as to what he can remove in terms of surgeries. We also have the difficulty of the blood-brain barrier, which is really, in certain terms, a really good thing, because it evolved to protect our brains from disease and from infections and things like that. But when it comes to treating things that go wrong in the brain, it makes it very difficult, because we have these specialised cells. So this is a blood vessel that feeds the brain, for example, and we can see this pink layer here is also diagrammatically shown here, and these blood vessels are very specialised. They're only found in the brain, and they make it very difficult for anything to penetrate into the brain, and that includes cells of our own immune system, which is why the brain has its own resident immune cells called microglia. But it also makes drugs very difficult to penetrate into the brain as well. A further complicating factor for brain cancer is what we call genetic heterogeneity. So basically within a spectrum of 10 different patients, no single patient will have the same kind of tumour at the level of their DNA. They all basically have a different kind of mutation that's developed throughout their life and that's caused their tumour. What they might have is some key, what we call driver mutations in common. So very common changes are in genes that we call tumour suppressor genes. So when they get mutated, a tumour can spontaneously form because they're no longer subject to the control that's usually given by these genes. But in terms of the tumour mass itself, no patient is identical, which makes finding a single treatment to cure lots of different people a challenge. And if you also go in and look at a single tumour in one of these 10 patients, what we often find is that there are patches of different disease within the same patient's tumour. So that what we call subclones. So this sort of pink patch of brain tumour tissue here will have a completely different set of mutations than this yellow patch of tumour here, and again this will be different from the blue patch here. So that again, that means if you have a drug or a treatment that's good at targeting the mutations in the pink patch, it might not necessarily be good at targeting the yellow or the blue patch as well. So again, one treatment does not fit all tumours. And what complicates matters even further is that you also have changes and diversity within the cells that make up the blue patch of the tumour and the yellow patch of the tumour and the red patch of the tumour at the genetic level. So what you can see here is a patient's cells that have been isolated and fluorescently tagged at the level of their chromosomes for a particular genetic defect. Some of the cells have the green defect, some have the red defect, some have mixtures of both. So again, that makes it very tricky to find a treatment that works. There's also the issue of cancer stem cells, which we believe at the moment are the cause of tumours themselves. So basically every tissue in your body has a small population, about 0.1% of stem cells that divide and replenish the tissues of each individual organ. When those stem cells get damaged in some way, they can then start to divide in an uncontrolled way, and we believe that that's what causes a tumour mass to form. And what happens is the main bulk of the tumour, if you think in terms of a beehive, is comprised of worker bees, the drone bees that go out and do most of the job of growing. But what the hive is initiated by is the queen bee of the hive, and that's what the cancer stem cell is. So the current treatments that we have, chemotherapies, radiotherapies and things like that, typically, not always, but typically only kill off the drones of the tumour, the worker bees of the tumour, but they always tend to leave behind the queen bee. And what happens is that while you get the bulk of the tumour being greatly shrunken and reduced almost to nothing, this queen bee cancer stem cell seems to sometimes stay behind and persist and can cause tumours to relapse, so they'll come back at a later date. So then what do we need to do better in terms of these difficulties that we know are inherent to brain cancer and the brain as an organ? We need to be able to target the cancer that we can't see or access in terms of surgeries, including disseminated disease that moves outside of the primary site of the core tumour. We need to be able to target these brain cancer stem cells that are driving the formation of the tumour and that are causing it to relapse because they're refractory to current treatments. We need to create therapies that are more potent, that can basically blast all the tumour cells away, including the cancer stem cells, but also be selective in the way that they blast so that they don't damage the surrounding healthy, normal brain tissue, which is sort of important. So then let's talk about some new treatment strategies that are trying to do these things. The first is to do drug screens of compounds that can selectively and specifically kill cancer stem cells. And what people and scientists are doing around the world, and particularly in Canada, there's a big program at McMaster University, is screening huge libraries of new drugs that are either from pharmaceutical creations or from natural compounds from trees and plants and things like that, and screening them to see if they can find drugs that can kill these cancer stem cells. They'll kill the queen bee, they'll we'll be able to kill the worker drones with current therapies like chemotherapy and radiotherapy, and that will basically mean that the entire tumour has been ablated and cannot relapse. But we also need to be careful that these drugs not only kill the cancer stem cells, but are selective in the way that they do it, so they don't kill any normal healthy cells of the brain. Another extremely promising area of therapy is called immunotherapy, and that basically means that you're getting a patient's own immune system to react against their tumour. So what happens in the body is that our immune system is really our homeland security. It keeps us safe from damage, and it keeps everything in working order. So if you have an infection, it comes in and it secures that infection down, it kills your virus or your bacterial infection, and you recover. And in fact, one of the key functions of our immune system is for cancer immunosurveillance. So our lifestyles expose us all the time to different mutagens that potentially could damage our DNA, and indeed do damage our DNA. From the foods we eat, to the environment we live in, to the radiation coming from space, we're exposed to these kinds of assaults all the time in our everyday lives. And what the immune system does, and in particular the T cells and the CD8 T cells, which are a subset of the T cells, do is to flow throughout your body and look for signs of these damaged cells that have become mutagenized in some way. And when they see them, they react against them and remove them from the system. They kill them and make sure that they're not a threat any longer. But obviously on rare occasions, this cancer immunosurveillance goes a bit awry. So when a sort of cancer cell is initially forming, there's this arms race between your immune system and the cancer cell. So the immune system sees your cancer cell and tries to attack it, but if your cancer cell has the right set of mutations, it can outpace the reactions of the immune system. And when it reaches a certain size, the cancer will actually begin to secrete immunosuppressive factors that shut down your own immune system. And so many cancer patients are in this immunosuppressed state. That means that their immune system can't react against the tumour without getting kick-started in some way. And so one of the options to kickstart a patient's own immune cells is to take their T cells out of their body and manipulate them in a dish in a way that basically equips them with the capacity to see the tumour as a target again. So they're no longer suppressed, they're activated again against the cancer. And this is called adoptive T-cell therapy. You then infuse those genetically engineered T-cells back into the patient, and they then react against the cancer and kill the cancer cells. This is a particularly good therapy for liquid tumours like B-cell lymphoma, because basically you have a single tiny cells, cancerous cells, flowing through the bloodstream, which is also where your immune cells are flowing that you've infused into the patient. And these single tiny cells are a really good target because they have a high surface to area ratio, and they can be targeted very easily from all angles by your immune cells. What happens in solid cancers like brain tumours is that there's big solid tumour mass. Instead of being single cells floating around, it's a huge mass of cells. And so this makes the penetration of immune cells very difficult, so that they can't penetrate into the very centre of the tumour. So what begins to happen is when the immune cell targets the outer edges of the tumour, which is all that it can reach just from a stereo conformational perspective, the tumour then reacts and evolves a way to grow without the target that those T cells are targeting. Cancers are very smart and very cunning, and we have to be very smart and very cunning to keep up with them. And one way we can do this is, in our lab, we're using viruses as ways to unmask tumours and basically allow your own immune cells, without any manipulation, to see the tumour as a threat. And so what the idea here is that you have a tumour cell, and you have your virus that only infects cancer cells. It doesn't infect normal cells because it's either naturally engineered that way or we've engineered it that way. So it infects the tumour cell, and through the course of its life cycle it replicates itself to produce lots of little progeny which bud out of the virus, and while it goes through its replication cycle it kills the cancer cell, it lyses it open. And once that happens, lots of danger signals spill out of the cancer cell. We have inflammatory signals, we have pieces of virus that are dangerous. They have this sort of mesmerizing molecular outfit that your immune system has evolved over millions of years to recognize. So the virus helps your immune system on this side see the tumour as a threat because it's infected with a virus now. And what happens is that the pieces of the tumour that are released when the tumour cell lyses open are recognised by specialised cells in your immune system known as dendritic cells. They recognise the pieces of tumour and then they present them to your CD8 T cells, which then react against the tumour. They also present them to other cells of your immune system which have direct abilities to lyse the tumour as well. So once you've started to infect a few tumour cells with your virus out of the entire mass, then the immune system basically does the rest and clears up any remaining tumour cells. And so in terms of the viruses that we use, we basically find them in nature, at least the basic viruses that we use. So viruses are all around us in the world that we live in. They live in the soil. So viruses replicating in the soil are an important contributor to the carbon cycle and to the ecosystem of the natural habitats. Viruses also exist in the air, and in fact we think that one of the main reasons that snow forms is that it finds a virus particle that acts as the core of the snow particle. So when snow's forming and falling, sometimes it's got a virus inside. Not a dangerous virus, don't worry about the snow. Viruses are also in the oceans, so when viruses replicate in the deep ocean sediments, they are an incredible part of the carbon cycle. And viruses also exist in living creatures. Most of the viruses in animals, insects, flies and humans, I guess we are an animal so I didn't need to say that, are benign and they cause no overt signs of disease. And indeed every animal has its own, what's called its own virome, which is basically the individual catalogue that each one of us has of the viruses that live in our bodies. Just like we have different bacteria in our guts, which is called our microbiome, we also have viruses that also help us as well. And lots of our genetic material, so in our own DNA, we have pieces of ancient viruses that we were exposed to in the historical epochs, and so the truth is that we coexist with viruses at all times. And in fact, the ancient viruses in our genome are driving our ongoing changes in genetic diversity, so they're helping us to evolve. And in terms of viruses being used as therapies, viruses are relatively simple organisms. So some have as few as 2 genes, which compares to humans that have about 24,000 genes. And if we understand the virus genes and a little bit about our own, then we can start to create viruses that can function as programmable machines. And in fact, we've already started to do that since about the 1800s, where we've used viruses to treat and protect against infectious diseases, and that's the basic concept behind vaccination, which is arguably one of the greatest inventions of our time. And one of the biggest vaccination success stories, if we wanted to have an illustrative example, is the use of infectious replicating cowpox as a basis for the smallpox vaccine. So the smallpox vaccine uses a virus to protect against a virus. Both viruses look a little bit similar, but cowpox causes a very mild, benign infection that's very quickly resolved. Smallpox doesn't. Smallpox kills us. So vaccination and using viruses to treat disease has led to the eradication of an illness that's basically plagued humankind since the ancient Egyptian dynasty. And really one of the first reports of viruses that could kill cancer started to be published in the 1900s and was first really got to the bottom of by the Romanian physicists Constantin Levaditi and Stefan Nicolaou, who in 1923 published a report showing that certain viruses replicated much better in tumour cells than they did in normal cells, and they postulated that tumours acted as a sponge to attract virus replication. And in fact, we now think that, or we now consider that oncolytic viruses, which are viruses that kill cancer, are obligate parasites of the tumour cells. Now in 1923 we didn't really have the tools or the science knowledge to understand why this was. We just observed that it was the case. But now we have a better grip on why it is that oncolytic viruses prefer to grow in tumours and don't grow in normal healthy tissue. And that's basically due to the sum of all the cancer mutations that a tumour cell creates in itself to solidify its own growth success. So there are probably 6 main things that cancer cells do to make themselves grow and be able to grow in a way that is not subject to the constraints of the normal immune system in the body. So one of the ways that they do this is to change the amount of stuff that's expressed on the surface of the tumour cell. And so they'll change levels of receptors, some will go up, some will go down, in order to grow better. And what viruses can do to exploit this is that some of those receptors that go up are actually the receptors that the virus uses to attach to the cancer cell. So this helps them to grapple onto a cancer cell compared to a normal cell. Another of the changes is that cancer cells typically increase their energy supplies and their metabolism. So basically when a virus is inside a tumour cell, it has access to an all-you-can-eat buffet of supplies to create its own progeny, which is basically what a virus really wants to do. It wants to replicate itself and create lots of little viruses. Other mutations that tumour cells undergo to secure their own success are decreasing apoptosis. So apoptosis is a form of programmed cell death that many diseased or infected cells will undergo in order to benefit the whole organism, because they're obviously going to endanger the body. But what cancer cells do is to take those pathways offline. So basically they're becoming almost immortal. They can grow and continue to grow as long as they have a nutrient supply. And what this means for viruses is that they also have, if they're infecting a tumour cell, an immortal home. They can live there for a long period of time, replicating themselves, creating their little virus progeny, and it forms a really good home for them. And so the sum of all these mutations that helps viruses to infect only tumour cells basically means that the viruses are tumour selective. So when we give the viruses into the bloodstream, they home specifically to tumour masses. They don't infect normal healthy cells, they only go to tumours. And this is demonstrated by a brain section here where the virus is expressing a fluorescent reporter and can only be seen in these 2 tumours here and here, but can't be seen in the normal healthy tissue outside the tumours. And this is seen here with a virus expressing a fluorescent protein, so you can see that the delineating line between the tumour and the normal healthy brain tissue is so tight. The virus is only in the tumour, it's not in the normal brain tissue at all. And so we know that the oncolytic viruses that we're using kill cancer cells in multiple different ways. They don't just attack one single cause of cancer, they attack lots of different ways. So they basically, as I said earlier, they'll get into a cancer cell and they'll burst it open, they'll lyse it open, and spill out all these inflammatory and viral proteins that the immune system can recognise and react against. So first of all, they blast open cancer cells, which is a good thing. Secondly, viruses replicate. So say if we put in 10 particles into a tumour, the virus will replicate and self-amplify that dose as it spreads through the tumour, which is good for the virus encompassing the whole tumour mass, so it can access all areas. We can also deliver the virus into the bloodstream and it will seek out and home to metastatic disease. So if you don't just have a primary tumour, but you have disease that's spread outside the brain as well, then it can home to these areas. It also can infect the malignant diseased blood supplies that the cancer has recruited to fuel its own growth. It can infect those what we call cancer-associated blood supplies, which they're not normal blood supplies, they're also malignant, considered malignant, so have to be dealt with. So it can do that, it can collapse down that blood supply and kill off those blood cells. The virus is also able to modulate the tumour microenvironment, so as I said, it gets into the tumour cell, spills out all these danger signals, and then the immune system can come in and react against it. And what's really nice about the fact that we're able to induce CD8 T cell responses against tumours and virus antigens alike is that these cells are responsible for immune memory. So when you get a vaccine and it lasts for up to 30 years, one of the reasons why is through CD8 T cells. They're long-lived and they remember what dangerous targets look like. So if you've got a virus that's induced an immune response for CD8 T cells against a tumour target, if that tumour recurs again at some period in the future, then they can also see that target again and prevent it from growing. So basically, it's a way to prevent your tumour from relapsing. So currently under development there are several oncolytic virus platforms. Most of them are for solid tumours for reasons I'll talk about in the next slide. So things like skin cancer, liver cancer, head and neck cancer. And the two main candidates in the most advanced phase clinical trials are vaccinia virus, which is from a company called Silogen, and it's marketed under the name Pexavec, and herpes simplex virus, which is from a company called Amgen, marketed under the name T-Vec. And like I said, these are in large-scale phase 3 and 4 clinical trials for solid tumours, but not really many are in trials for brain cancer. And the reason for this is that most current oncolytic viruses fail to hit that sweet spot that balances safety in the brain and anti-tumour killing activity. So for example, the T-Vec from Amgen has really good safety in the brain, but it doesn't do that much against the brain tumour itself. Conversely, vaccinia virus, which was the Pexavec that's in large-scale clinical trials, has really good anti-cancer activity. It blasts through the tumour, but it has poor safety in the brain. It has unwanted side effects in the normal healthy areas of the tissue. So it's almost too good for the brain. There are, of course, exceptions to that rule. So one virus that is having good success for brain cancer is based on poliovirus. It's called PVS repovirus. It was created by Matthias Grummeyer at Duke University in the US. And so far he's only treated a small number of patients. He's treated 5 patients so far with glioblastoma that's recurred. So none of these oncolytic viruses have really been used as a primary treatment. It's always been sort of a secondary treatment when the tumours actually recurred. But he has seen excellent responses in one patient where there's basically been a durable response and the tumour has regressed very nicely. So this was 2 months after treatment. This is the tumour mass here, which is considerably shrunken from pre-treatment, and 9 months later the tumour has continued to shrink even further. And this was in a 16-year-old teenage girl, and she's now back at college and is continuing with her studies after this treatment. 3 of the other patients also had very good responses. They didn't have durable cures, but their tumours did shrink greatly and they prolonged their survival. So what we're interested in doing in the Stojdl Lab at the CHIO Research Institute is to try and find viruses that tip this balance towards finding a candidate that's safe in the brain and has excellent anti-brain cancer activity in the brain. So basically we've concentrated our efforts on a family of viruses called rhabdoviruses. These are bullet-shaped envelope viruses that are about 180 nanometers long, very, very small. They replicate exclusively in the cytoplasm. They never penetrate into the nucleus of the cell, which is where the DNA is. And they have about 5 genes, which is encoded by 11 kilobases of DNA in an RNA genome. Rhabdoviruses are not human pathogens, so there's no risk of any dangerous unintended consequences. Actually, most of them cause, or some of them cause, a self-limiting disease in cattle, which is spread by an insect vector between cows. And we have basically gone to a biobank at the University of Texas, where they have many different viruses held through the career of Robert Tesh, who's an environmental researcher that's gone through the world and isolated viruses from different organisms, and we focused our efforts on 7 different viruses that were available in that biobank, all rhabdoviruses. 3 of them were isolated from the US, Muir Springs, Farmington and Bahia Grande. 2 were isolated from Brazil, Caracas and Maraba virus, from sandflies, and 2 were isolated from Australia, Tibragargan and Nagangan virus, from ticks out in Australia. And so what we did was to, somebody else had done the bioprospecting part of this, which was very fortunate. We didn't get the excitement of going all over the world and finding these viruses, but we did screen them. So for those 7 viruses, we tested how effective they were against different cancers, and we were particularly interested in finding ones that were effective against cancers of the central nervous system, like brain cancer. And what we found was a very variable spectrum of how good these cancers were at killing, these viruses were at killing cancer. So some, like the 4 on the left, weren't very good at killing any kind of cancer, but the 3 on the right were excellent cancer killers. And in particular, we were interested in Farmington virus, which could kill 100% of tumours of the central nervous system. Now, Farmington virus looks like this under the electron microscope. It's got the typical bullet envelope shape of its brothers and sisters in the rhabdovirus family. It actually segregates into the genus Cytorhabdovirus. And what we wanted to do was basically to test it even further on more cells from isolated from brain cancers, to really find out if it could kill them and if it was worth pursuing as a therapy. So what we did was to compare its activity on normal brain cells, GM38, and normal human astrocytes versus brain cancer cells, 3 cell lines we've got there. And what we found was that it was safe in normal cells, because basically we're looking at survival of the cells here at increasing doses of virus along this axis. And what we see is that the normal cells, which are in blue and turquoise here, don't start to die until you have an incredibly high dose of virus here. So at all lower doses of virus, they're not affected by the virus itself. Whereas the tumour cells start to die very quickly at relatively low doses of virus. So this is 0.0001 virus particles per cancer cell. And that's the number of viruses that you begin to start to see effects. So that's actually very impressive. So we would consider Farmington virus to be hypervirulent on these tumour cells, but safe and attenuated on the normal cells. And we also looked to see how Farmington virus could be safe in the brain, because most of the brain in a cancer patient is not replicating. So the cells are basically in G0 of the cell cycle. So most cells in the body are proceeding through the cell cycle and proliferating and dividing. But what we see in the brain is that most neurons are in the G0 phase. They're quiescent and quiet. They're just hanging about, doing their job, and not dividing. And that's actually a really good thing for Farmington virus, because that means that the only thing that's dividing in a brain of a cancer patient is the tumour. And so what we find is that comparing untreated cells that are either going through the cell cycle and dividing in grey versus ones that are not dividing in purple, there's no difference in how these cells survive after no treatment. But when we add Farmington virus, the only cells that die are those that are proceeding through the cell cycle, so ones that are proliferating and dividing, and cancer cells. So that's a really good safety feature of Farmington virus. We also find that Farmington virus kills tumour cells through apoptosis, which is a form of cell death. So what we find in an untreated tumour is that there is a basal level of apoptosis, which is basically, I said to you earlier that most tumour cells were considered immortal. But that's not entirely true, because when a tumour mass is dividing, it needs the blood supply to keep up with it. So if it's dividing too quickly and the blood supply is not bringing in enough nutrients and energy, oxygen, and taking away enough of the byproducts of growing, then the tumour cell can't survive. And particularly what we tend to find is that at the middle of a tumour is where there's actually quite a lot of death going on, because the very central part of the tumour mass is this area that the blood vessels just can't penetrate. But however, even though we have a basal level of apoptosis, when we add our virus, you can see that the signal for cell death, which is in red here, goes through the roof. So the virus is causing a huge amount of cell death in the tumour cells itself. And we can see that the virus infection is in green here, and where the virus is where the cells are dying, which is what you can see on the merge here. So the green and red combine to make a yellow signal. So we know that it's the virus that's causing that death. I also mentioned earlier what a problem brain cancer stem cells were with most traditional therapies not being able to target them. What we find is that Farmington virus can get into brain cancer stem cells. So these are 3 different patients that were kind enough to donate part of their biopsies to our research and allowed us to test our therapies in these important cell populations, which we believe are why current therapies fail. So we have 3 patients, and we have the virus infection shown in green. So basically the virus gets into every brain cancer stem cell, which you can see in the phase contrast images here. It gets into every cell, it kills about half of them. So we have 3 representative patients here. Patient 8, the virus kills them very, very effectively at a very low dose of virus. Patient 7, you need a slightly higher dose of virus to start killing these brain cancer stem cells. In this patient, you kill all of them. In this patient, you kill about 25% of them. And in this patient, we're not doing that well with these brain cancer stem cells. So there are still ways that we can improve our virus, we think, to actually attack greater levels of the cancer stem cells. But we're not doing too badly. 50% is pretty good, and it's more than current therapies do at the moment. So we've done these screens, we've found that our oncolytic Farmington virus doesn't kill normal brain cells, but it does kill malignant brain cells. So we've sort of managed to identify rhabdovirus as an ideal family for producing these oncolytic virus candidates, and we've identified Farmington as potentially a really good brain cancer candidate therapy. So now what we want to do is to take our research out of a dish and into an animal, and basically check that we don't cause any unintended side effects in a normal brain. And so we tested this out in a normal mouse brain, and we injected the virus directly into the brain as an intracranial therapy at a very high dose. So we wanted to make sure that the highest dose that we'd be using in a clinic was safe in our models. And so what you can see here is VSV, which is a virus that cannot be used in the brain. It kills normal brain cells with as little as 2 virus particles. And you can see how damaging VSV is in the brain. These areas of damage and necroptosis, cell death is shown in the inset here. You can compare that to an untreated mouse brain, which shows a lovely amount of neurons, they're very healthy, and actually our Farmington treated animal looks exactly the same as the untreated control. So Farmington virus does not have any unintended off-target effects in a normal, healthy brain tissue. And so then we wanted to move on to test if the virus actually worked against brain tumours. We've seen that it does it in a dish with human brain tumour cells isolated from tumours, but we wanted to see if it can do it in a living animal, because that's ultimately what we care about treating. So we tested our Farmington virus in 3 different mouse models. We have a glioblastoma, a glioma, and an astrocytoma. And this one is the most difficult to treat. It's very aggressive and hard to treat. So that was the one that we're potentially most excited about. So we had 3 different groups of mice treated with 3 different things. One was the control, which is just getting an infusion of saline into the brain. One was giving Farmington virus directly into the brain, so intracranially, and one was giving Farmington virus into the bloodstream, so intravenously injecting it. And what we found in the glioblastoma model is that we can cure about 20% of mice in a long-term fashion when they get Farmington virus in the bloodstream. In the glioma model, what we see is that we can cure about 18% of animals when they get Farmington virus into the brain. And in the really difficult to treat mouse model, CT2A, an astrocytoma brain cancer, what we see is that we can cure between 20 and 30% of mice when they get virus either directly into the brain or through the blood supply. And the reason that we're testing blood supply versus intracranial methods is that it would be easier if we could find a treatment that could be just injected into the blood supply. It just would be an easier thing for patients not to have to undergo not only a surgical resection, which requires you to penetrate into the brain, and then have to get this virus also penetrating into the brain at a sort of secondary point in time. So it would just be more practically applicable if we could have a virus that could be given just intravenously. And so if we summarise these GBM studies, not just in these 3 models, but in other mouse models as well, what we see is that the number of responders that we're getting, so basically where we see that the tumours regress and shrink, we get around 70 to 80% of responders doing that. In terms of durable cures, in terms of durable cures, what we see is that we get, when we give the virus through the bloodstream, we get about 30% of mice that are cured reliably of their tumour. And when we go in and challenge these mice again, when they've cured their tumour, we go in 6 months later and give them their tumour back again, they reject it. So basically what that's saying is that our virus has managed to generate immune responses and immune memory responses that remember what the cancer looks like and react against it if they ever come into contact with it again, which sort of mimics the situation in a patient where you'd have this relapse and recurrence of tumour. Our virus can deal with that. So Farmington virus also improves survival, at least as well as current best treatments, temozolomide and Avastin. So what you can see here is that in untreated animals in black versus mice that receive temozolomide, there's a really nice extension in survival time, but nobody gets cured. In terms of Avastin, the untreated is in black, and then the highest dose of treatment is in blue. So again, you get a really nice extension of survival, but you get fewer animals actually being durably cured. So you don't really see any long-term cures in these same mouse models treated with existing gold standard treatments. And the use of our oncolytic virus has important implications for pediatric cancer. So I said that brain cancer is one of the most common solid tumours that's seen in children, and one of the issues with using chemotherapy in children is that it has unintended consequences down the road that might not necessarily be as much of an issue for an adult. So in the New England Journal of Medicine, they put it particularly well. They said that although the cure rates of childhood cancers appear to be very good with existing treatments like chemotherapy, actually 60% of the children that are cured of their cancers using these techniques will go on to suffer significant side effects as a result of that therapy, and sort of 30% of them will contract secondary cancers that are as a direct result of the chemotherapy treatment itself. So basically what that means is that the chemotherapy, not only does it damage the DNA of the tumour cell, which is what we want to damage the DNA and kill that tumour cell, but it also can damage the DNA of normal cells, and that means that at some point in the future those cells might go on to form a tumour of their own. And one of the great strengths of oncolytic viruses is that they do not damage the DNA. So all the replication cycle of oncolytic viruses goes on outside the nucleus in the sort of blue area of this cartoon cell here. The oncolytic viruses never penetrate into the nucleus of the cell, which is where the chromosomes and the DNA, the genetic material, is held. So they do not have any of these genotoxic consequences that chemotherapies do have, that are particularly debilitating in childhood cancers. So to summarise, brain cancer therapy is a very challenging area, but the clinical toolbox of therapies that are available for brain cancer treatment are rapidly expanding. There's an incredibly intense amount of research going on right now across the world, and Canada's at the forefront of it, but like I said, there are labs in the US and the UK, across Europe, that are all focused on expanding the number of therapies that are available for brain cancer patients. And these therapies are getting more sophisticated to meet this challenge, so they don't just go in and kill lots of different cells and don't differentiate between the tumour and the normal cells anymore. They actually go in and selectively kill the tumour cell. which is really what we need to help long-term cures. Early results with our own Farmington virus, as well as other oncolytic virus platforms in development in labs across the world, are looking very promising. And there's still great potential, though, for new innovation and development of these oncolytic virotherapies and other therapies as well. In terms of clinical trials, we're hoping to get our Farmington virus into clinical trials within 2 years. So that's the stage that we're at now. We're currently trying to secure funds for making that happen. So we're actually going to start a Kickstarter campaign to get the public support for making our promising platform a reality. In the meantime, though, there are a lot of cancer clinical trials going on for brain cancer specifically. I don't know of any equivalent database in Canada, but this is from America, which is at least on the same continent. And this is a really useful database hosted by the National Cancer Institute and the National Institute for Health. And they basically have compiled a list of every clinical trial that's active or recruiting, and you can go on there and search your particular cancer type and condition and find clinical trials for your disease. And this is the website here. And actually some of the trials within Canada do come up on this website as well. So it's a really good resource for anybody that's interested in looking to see what else might be out there beyond gold standard treatments. And so our lab, the lab of Professor David Stojdl, is online, so we post lots of updates about our own research and about clinical and oncolytic virus research across the world. So we're online at stojdllab.ca. We're also on Twitter and Facebook, Google Plus and Tumblr. And so our lab is comprised of my boss, David Stojdl, as well as Melanie Labelle, Charles Lefevre, Christina Allen, Stephen Baird, Caitlin Rowe and Beta Yadolai. And that's our team at CHIO. And it just remains for me really to thank our funders who, without their support, none of this research would have been possible, and particularly Brain Tumour Foundation of Canada, who's a really good supporter of our research. Thank you.